Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, in previous class, we have already talked about the structure of DNA in molecular biology and about their discovery and the components of DNA that all we have discussed and different types and the properties also we have discussed. Today we are going to talk about the central dogma, then the packaging in prokaryote and eukaryote. So first, if we talk about a central dogma of life, central dogma means it is a central thing actually which is always responsible for the anything which is occurring in our body or in life. Then we call it as a what, central dogma of life. And this central dogma given by a Francis Crick, proposed by a Francis Crick in molecular biology, and this states that or explains one way or unidirectional flow of information from a master copy of DNA to a working copy RNA. It is a flow of information from a DNA to a RNA and from RNA to a building molecule or a trait expressing molecule, that is polypeptide. So here you can see DNA to mRNA to the protein. So if DNA get copied, then it is known as replication. The flow of information from DNA to mRNA, then it is known as transcription, and from mRNA to protein, that is known as translation. Okay, so this is known as central dogma of life. Now, but this happens when the genetic material is DNA, but in retroviruses, RNA act as genetic material. And if we take example of retroviruses, HIV, in this the flow of information is in reverse direction, that is from RNA to DNA. And when this reverse flow of information occur, then it is known as reverse central dogma of life. And the reverse central dogma of life, if you call, it is also known as feminism, because it is a exception to the one way flow of information, or we can say a central dogma of life. And this is reported or proposed by Temin and Baltimore. It is proposed by Temin and Baltimore, and they they independently discovered reverse transcription. Reverse transcription means formation of DNA from the RNA in some viruses. And these viruses produce an enzyme reverse transcriptase, which can synthesize DNA over the RNA template. So if we talk about reverse transcription, here we write DNA to mRNA, then mRNA to protein. Now DNA to DNA is a copying of DNA, then it is known as replication. When DNA form RNA, it is known as transcription, whereas formation of DNA from the mRNA, it is known as reverse transcription. And when protein is formed, it is known as translation. Okay, and this translation will lead to the formation of protein from the mRNA. So it is a reverse flow of information. It is known as reverse central dogma. And this discovery was important in understanding cancer, and hence these two scientists was awarded Nobel Prize for this. and this modified flow of information can be seen like this. So this is the reverse central dogma, which is also known as feminism. And in this reverse transcription, if we talk, it is done by enzyme reverse transcriptase. Next, after this, there is a packaging of DNA in DNA helix, or we can say a DNA strand can be packaged. Suppose because we know that DNA is too much long and it is packaged in a nucleus of a single cell. How it is possible? So that is helped by the some of the proteins. So if we talk about packaging of DNA helix, if we take a distance between two consecutive base pair, that is 0.34 nanometer, or we can say 0.34 into 10 raised to minus 9 meter. Now, the length of a DNA double helix is atypical typical in a typical mammalian cell if it is calculated simply by multiplying the total number of base pair with the distance between two consecutive base pair. That is 6.6 into 10 raised to 9 base pair are a base pair in a diploid cell into 0.34 into 10 raised to minus 9 meter per base pair. That is our distance between two base pair. Then it turns out to be approximately 2.2 meter long DNA. So in a diploid set, the length of the long DNA is how much? 2.2 meter. Now, this length is far greater than the dimension of the typical nucleus because approximately the dimension of a nucleus is 10 raised to minus 6 meter. So, how is such a long polymer packaged in a cell? That is one of the important question here. So, here answer to the question can be obtained in this topic, that is a packaging. Okay. So the length of eukaryotic DNA is far greater than the dimension of a typical nucleus. Nucleus dimension is represented 10 to minus 6 meter we have discussed. So the DNA of prokaryote and eukaryote packed in a space which is 1000 to 10,000 times smaller. Our size of nucleus is 1000 to 10,000 to 1 lakh times smaller, and such packing occur differently. So packaging, if we talk in case of prokaryote, in prokaryotes such as E. coli, though they do not have a defined nucleus, DNA is not scattered throughout the cell. In prokaryote, if we talk about the E. coli, there is no defined nucleus, but DNA is not scattered throughout the cell. DNA is obviously defined in a central position. So here, DNA being negatively charged is held with some protein which have positive charge in a region termed as nucleoid. So in prokaryotes, what happened? The DNA found in cytoplasm is in supercoiled state, and the DNA nucleotide organize large loops held by a protease. And the DNA of prokaryote, if we talk, in prokaryote there is no nucleus, almost circular, and lack chromatin organization. So this is referred as genophore. Okay. Now, for the packaging in case of prokaryote, this supercoiled stage, if we talk, supercoiled stage of this nucleoid is maintained by non-histone basic protein, non-histone basic protein, that is polyamines, which have positive charge. Okay, so the non-histone basic protein are used here, which have positive charge for that are known as polyamines for the coiling of this DNA in a nucleoid, and this structure will be referred as a nucleoid or genophore. So here non-histone protein are used in case of prokaryote or bacteria. But when we talk about the DNA packaging in case of the eukaryote, dear student, here different type of proteins are used. In eukaryotes, this organization is much more complex, and this packaging is also more complex. Here, set of positively charged proteins are used, which are basic protein, and they are called as histones. So, if we talk here, the histone protein, this histone protein are like this. Okay, and in histone protein, you will see that this is a H1 protein, and this is a particularly histone octamer. Okay, so if we see, suppose this is a chromosome. Okay, and this is a one chromatid, two chromatid are forming chromosome. This is centromere region. If we stretch a DNA from this chromatid, you will see that it will separate and it will form a chromatin fiber. When it will more decondense, you will see the chromatin. Then in a more decondensed stage, you will see the nucleosome, and in more decondensed stem, you will see the what, a DNA double helix. Means if we package DNA double helix, we are going to see the formation of nucleosome, linker DNA, or we have chromatin content, then chromatin fiber, then chromosome, and like this coiling is going to be happen. And for this histone protein are going to be used. Now the structure of histone protein, if we talk, histone protein is made up of histone octamer and H1 protein. This is a core of 8 histone. It is a histone octamer. And here, four types of polypeptide or protein are used: H2A, H2B, H4, and H3. And these four proteins are present in dimer form or two subunits. So here also at the back side, you will see the H2A. This is also H2B. This is H3. This is H4. Okay, these are found. And this whole histone is coiled by DNA. When DNA coil around this octamer, DNA takes 1/3 turn around histone octamer, and there is a H1 protein here which is present like this. When H1 protein attach with this histone octamer, it is going to form a nucleosome structure. Okay, so in eukaryote, this way histone octamer is helpful in particularly this. And these histone are also rich in basic proteins or basic amino acid, that is lysine and arginine. Okay, and the five types of histone protein are found, that is H1, H2A, H2B, H3, H4. Four of them occur in a pair, they forming a histone octamer. Dear student, this histone octamer is also known as a new body. Okay. Now, negative charged DNA is wrapped around a positively charged histone octamer to form a structure called a nucleosome. Now, in this DNA also, which turn around takes 1/3 turn around histone octamer, and it is this DNA is of 200 base pair. Okay. Now, when further coiling continues, what will happen? Suppose in further coiling continues, suppose this is the DNA, and DNA have a diameter of 2 nanometer. Now, if further coiling occurs around histone octamer, there is a 10 nanometer structure is formed, which is known as nucleosome. It is 10 nanometer structure. Further coiling occurs, it is going to form a solenoid, and solenoid is 30 nanometer structure. It is a further coiling of this octamer containing like this, that is solenoid or 30 nanometer. Then it will further coil on, and you will see the further coiling form a super solenoid. structure which is a 300 nanometer, and the further coiling continues, it is going to form a chromatid that is 700 nanometer structure, and chromatid is going to look like this. But when chromatid replicates, it is going to form a chromosome like this, and this chromosome is going to form a 1400 nanometer structure. Okay, so like this, whole packaging of DNA occur and a chromosome are going to form. And for this, basic proteins are used, that is a histone protein. Now here, histone protein is used, but apart from histone protein, non-histone chromosomal protein, non-histone chromosomal proteins that are NHC are used in packaging of chromatin at higher level. And this NHC are of three types. Number one, scaffold or structural non-histone chromosomal protein. Second, functional non-histone chromosomal protein. Now, functional non-histone chromosomal protein include DNA, polymerase, and RNA polymerase. And third include regulatory non-histone chromosomal protein. And the example include HMG, that is high mobility group protein that control gene expression. Okay, so these three are the non-histone chromosomal protein. And when the chromosome are formed, when we talk about the chemical composition of chromosome, this chemical composition of chromosome include DNA, that is 40%, RNA, that is 1.2%, histone protein 50%, acidic protein are 8.5%, lipids are in traces or very small amount, and calcium, magnesium, and iron are also in traces, that is in a very low amount. Okay, So this thing is seen in case of the chemical composition in a chromosome. Now, if we talk further, then a protein acquire a charge depending upon the abundance of amino acid residues with its charged side chain. And histone protein are used, and histone protein have arginine and lysine, basic amino acid, positively charged basic amino acid, which gives them basic nature. Histone are rich in basic amino acid, that is the lysine and arginine. Both the amino acid residues carry a positive charge in their side chain. And when this histone DNA wrap around the histone protein, it is going to form a nucleosome. Histone are organized to form a unit of 8 molecule called a histone octamer. And histone octamer wrap around DNA wrap around the histone octamer and form a nucleosome. So the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called a nucleosome. And you can see this is the nucleosome, where you can see this is the octamer and DNA is coiled around this. We have discussed that this DNA which connects the two octamer, that is known as linker DNA. Now, on one nucleosome will act as like a bead which consists of 8 histone molecule plus approximately 146 base pairs of DNA, or approximately 200, up to 200 can be there, and gives a 10 nanometer DNA structure. Okay, so histone protein is like this and DNA will wrap around like this and the nucleosome is going to form. A typical nucleosome contain 200 base pair of DNA helix. Be remember, typical nucleosome contain 200 base pair of DNA helix around the histone octamer. And nucleosome constitute the repeating units of structure in nucleus called a chromatin. Nucleosome will wrap and they will form a structure which is known as chromatin. The nucleosome in chromatin are seen as a beads on a string. When we see the chromatin fiber, that is a solenoid structure, or we can say uh, solenoid, or we can say 30 nanometer structure, which is also known as chromatin. That chromatin is formed by the coiling, further coiling, and the nucleosome in chromatin, if we take a C, they are seen as beads on a string when viewed under the electron microscope. So this is the histone octamer, and over which the DNA will coil like this. The beads on a string structure in chromatin is packaged to form a chromatin fibers. Further, chromatin will package and form a chromatin fiber that is further coiled and condensed in metaphase of cell division to form a chromosome. So the further coiling is going to form a chromosome, and the packaging of chromatin at higher level require additional set of protein that is known as non-histone chromosomal protein. Whatever the non-histone chromosomal protein are used, which we have already discussed. In a typical nucleus, if we see the chromosome is formed, and when we stain this chromosome, you will see that the chromatin hair is, some regions are darkly stained and some regions are loosely stained or lightly stained. So in a typical nucleus, if you see, some region of chromatin are loosely packed, so enzymes can interact them with them, and these are lightly stained because they are not tightly packaged, and that is known as euchromatin. The chromatin that is more densely packed and stains dark, it is known as heterochromatin. So heterochromatin is tightly coiled and euchromatin is loosely coiled. Now euchromatin is loosely coiled, so enzyme can interact easily with the DNA. But in heterochromatin, an enzyme cannot interact with the DNA because it is tightly coiled. So euchromatin, if we consider, it is a transcriptionally active chromatin. Whereas if we talk about heterochromatin, it is transcriptionally inactive. Okay. So this is the thing which is seen in case of the particular packaging of this DNA. So before discussing MCQ, let's talk about one question. And if we discuss question, if a DNA molecule has 2000 base pair, then calculate the A, number of sugar and phosphate residues, sugar and phosphate molecules, and B, number of N glycosidic linkage. If we talk about number of sugar and phosphate molecules, here if we talk, here 2000 base pair is there in DNA. So total number of nucleotide are 2000 into 2, that is 4000. And nucleotide is made up of nitrogenous base, sugar and phosphate, plus sugar and phosphate. So here this 4000 nucleotide are there. So obviously the number of sugar is equal to 4000 and number of phosphate they are also 4000. Next is number of N glycosidic linkase. Now this glycosidic linkase occur in between nitrogenous base and sugar. So total number of N glycosidase linkase will be equal to number of nucleoside. So 4000 is going to be the answer here. Okay. Now after this, let's discuss another question. DNA was extracted from Streptococcus bacterium. The proportion of adenine was found to be 28%. Then calculate the amount of cytosine. So if we talk here, then according to Chargaff's rule, A is equal to T. So here, if adenine is given is 28%, then thymine is also 28%. So it is give rise to the 56%. Now we need to remaining identify cytosine. So guanine plus cytosine if we talk, because they both are in equimolar concentration, that is 100 minus 56, so that is 44%. So guanine is equal to 22% and cytosine is equal to 22%. So cytosine is going to be 22% if the DNA has a adenine has a this much. Okay, so this is the thing how you can solve this type of question. So let's discuss some of the questions. Histone are rich in basic amino acid like that we know very well, arginine and lysine. So second is going to be the correct answer. A typical nucleosome consists of 200 base pair of DNA helix around a histone octamer, and that is second answer. Okay, dear students. So this is all about the central dogma, reverse central dogma, and packaging of DNA. Thank you.